the CCR5 co-receptor, the virus cannot infect the cell. So you're essentially immune to it, aren't you? So there is a small percentage of the population that will be immune to the virus. That's the case with all viruses, if you think about it. All new viruses that come into the population, into the community, even if it's one that has a high virulence and a lot of people are susceptible to it, there's always going to be enough variation in the population, especially the size of the human population, that a few individuals are going to be missing the surface receptors that that virus or that bacterium might use to infect those cells. So it's unlikely that we'll find a virus that wipes out the entire human population. It could wipe out a large number of us, but not all of us. So now, the virus is bound to the co-receptors. That induces then, as these co-receptors pull this transmembrane molecule, will pull the virus so that their membranes now fuse, and the virus deposits the protein cap into the cell, and the protein cap then disintegrates, releasing into the cell the enzymes that are associated with it, in addition to the RNA molecules. Now, reverse transcriptase. This molecule here is extremely important. What it does is it allows the RNA molecule to be transcribed into a DNA molecule. So what happens is that this reverse transcriptase will copy a strand of DNA using the RNA molecule as a template. Okay? Then this will separate into the two strands, and the second strand here that is DNA, this is DNA now, gets copied again by reverse transcriptase using this DNA molecule now as a template. So now we end up with a double-stranded DNA molecule. So reverse transcriptase takes the RNA molecule of the virus and ultimately converts it into a double-stranded DNA molecule. Over here is our nucleus, and we have our chromosome in here. Now, integrase comes in, and it'll pick up this double-stranded DNA molecule. It modifies it a little bit. It cuts off a couple of ends here to make them sticky. I won't ask you that necessarily, but cuts those ends off, and then it carries this double-stranded DNA molecule into the nucleus, where integrase will clip out a section that then will allow for the viral, using purple now, to be integrated into the host DNA. So now the viral DNA is in the host DNA. So every time this cell makes a copy of itself, the new cell is going to have a copy of the virus. But what happens generally is that during the infection, the virus is able to take over the machinery of the cell. And so it starts making copies of its DNA. Not making copies, making messenger RNA molecules. Now, if you remember, messenger RNA molecules will be synthesized into proteins, And what happens here is as we make our messenger RNA molecules, we are essentially making all of the parts that the virus needs. The enzymes that it needs, copies of the RNA, the capsid that will be needed, the surface protein that will be needed. So these are all being made. You got lots of stuff accumulating. Yes? Was there a question? Oh, I thought I heard a question. So you've got all this stuff, all the enzymes, messenger RNA, capsid molecules, all being produced. And the last protein, protease, plays the role in pulling these things together, clipping them up where they need to be clipped, because some of the proteins are going to need to be modified, so it modifies those proteins. And what will happen is that on the back side here, as these get pulled together, you'll see that the capsid will form with the RNA molecules, with everything else inside. While all this was going on, the other thing that was happening was we were studying the surface with our surface molecules. So imagine now the surface of the cell is studded with viral molecules. So now when this virus pushes out through excitosis, we're going to end up with a virus that is ready to go out and infect another cell.
So the protease is acting in the cytoplasm, correct?
Yes, it is.
Is the protease actually creating the capsid?
No, it's not creating the capsid. What the protease is doing is these proteins that are being produced here via the viral DNA, they need to be clipped up. You know, they need to be sized and put together and stuff. And the protease is really serving to size all of those proteins. So it clips them all up. Not really sure what pulls everything together, but there's a factor just underneath the cell membrane that kind of pulls all of the components together such that the virus, the capsid forms with everything in it, and then it escapes. Something to realize here, mistakes are going to happen in this pulling together. So there will be times when a virus might form that doesn't have everything it needs. And then that virus might not be able to infect another cell. But, go ahead.
Is that also a way of mutating when that virus does that? Have they found mutations in HIV yet?
All the time. All the time. But the mutation isn't happening over here. The mutation is happening here. Okay, so anytime you're making a copy of yourself here, you can cause mutations to occur. There are 20 different subtypes of HIV, so there are a lot of different strands of HIV. Any questions? So let's talk about some of the ways that we can control the infection. First off, AIDS is passed via sexual intercourse, exchange of blood, hypodermic needles. So you've become exposed to the virus as it moves through the circulatory system. If you can block any of these enzymes from being able to carry out their function, whatever it might be, you can prevent the spread of the disease. That's what a lot of our medications do. You've heard of AZT, maybe one of the first medications. involved in interfering with the replication of the virus. Now we have cocktails, which are actually 3 different medications. See, the virus has a tendency to mutate such that AZT lost its effectiveness after a few months to a year of treatment. So they had to introduce a new drug, DDI, and then it lost its effectiveness after a while too. So they have to alternate and change the drugs to keep them effective. It used to be that when you came down with AIDS, it was pretty much a death sentence. Within about 10 years, most people would have succumbed to the disease. Now it's not, because of the drugs that we have. Magic Johnson, who came down with the disease in 1990, is still doing quite well. Now Magic Johnson's got a lot of money, and he could afford the drugs that were being developed. But if you're infected, if you can block the receptor sites, so we've developed drugs designed to block CD4 or designed to interfere with the co-receptor. So if you can block that, you can prevent infection. We've developed drugs that interfere with reverse transcriptase. So if you can prevent the protein from copying the RNA to the DNA, you can prevent or slow down. Recognize what we're really doing, we're just slowing down the spread. You can interfere with integrase so that it can't carry the DNA into the host nucleus, or you can interfere with its ability to integrate it into the DNA. You can interfere with protease. And so we've got drugs that will interfere with all of these steps here. But it's still not enough to prevent the spread. I mean, the microbe is still able to move from one cell to another cell. Partly because you're talking about hundreds of thousands of viral molecules every day being formed.
I have a question.
Sure.
If you're interfering with CD4, how does that, if you have a drug that you introduce that interferes with CD4, how does that affect the rest of the cell's function?
Doesn't seem to affect the rest of the cell's function. We don't know what the CD4 receptor site is being used for. Maybe it isn't being used at all at this time. You know, we're not really clear what its function is. So, a couple of videos I want to show you here. Hopefully they come up.
Entry of HIV into the cell requires the presence of certain receptors on the cell surface. CD4 receptors and co-receptors, such as CCR5 or CXCR4. These receptors interact with protein complexes, which are embedded in the viral envelope. These complexes are composed of two glycoproteins, an extracellular GP120 and a transmembrane GP41. When HIV approaches a target cell, GP120 binds to the CD4 receptors. This process is termed attachment. It promotes further binding to a co-receptor. Co-receptor binding results in a conformational change in GP120. This allows GP41 to unfold and insert its hydrophobic terminus into the cell membrane. GP41 then folds back on itself. This draws the virus towards the cell and facilitates the fusion of their membranes. The viral nucleocapsid enters the host cell and breaks open, releasing 2 viral RNA strands and 3 essential replication enzymes: integrase, protease, and reverse transcriptase. Reverse transcriptase begins the reverse transcription of viral RNA. It has 2 catalytic domains: the ribonuclease H active site and the polymerase active site. Here, single-stranded viral RNA is transcribed into an RNA-DNA double helix. Ribonuclease H breaks down the RNA.